Welcome to aminacademy.com. In questions, how, why, and what. In that, we'll ask some question. Is genetically modified human possible? Modification of human genes in order to treat genetic disease is referred to as gene therapy. Gene therapy is a medical procedure that involves inserting genetic material into a patient's cell to repair or fix a malfunctioning gene in order to treat hereditary illness. Now let us discuss further detail about genetically modified human possible or not. Yes, genetically modified human is possible with technologies like CRISPR, allowing for precise editing of the genome. But the ethical and societal implications are complex and require careful consideration. What is genetic modification? Genetic modification or gene editing involves altering an organism's DNA to change its traits or functions. Techniques like CRISPR, clustered regularly interspaced short palindromic repeats, enable scientists to edit DNA with high precision. This technology has the potential to correct genetic defects, treat diseases, or even enhance certain human capabilities. Current applications and future possibilities. Therapeutic uses. Gene editing is currently being explored for treating genetic diseases like sickle cell anemia and cystic fibrosis. Somatic versus germline editing. Somatic editing involves modifying cells in a specific part of the body, and the changes are not passed down to future generations. Germline editing involves modifying germ cells, sperm or egg, and the changes are inheritable, meaning they can be passed down to future generations. Ethical and societal concerns. Germline editing raises significant ethical questions about the potential consequences for future generations, the possibility of unintended effects, and the potential for misuse. There are concerns about the potential for genetic discrimination, the creation of a designer baby scenario, and the unequal access to these technologies. Regulatory frameworks. Many countries and international organizations are developing guidelines and regulations to ensure the responsibility and ethical use of gene editing technologies. For example, the World Health Organization WHO has issued guidelines on human genome editing. Now, examples of gene editing technologies: CRISPR, case 9, a widely used gene editing tool that allows scientists to precisely cut and modify DNA. Other gene editing technologies. Researchers are exploring other methods for gene editing, such as zinc fiber nucleases, ZFNs, and transcription activator-like effector nucleases. Now, further, is genetic modification of human good or bad? Human genetic modification presents both potentially benefits and risks, with the possibility of treating diseases and improving quality of life, but also raises ethical concerns and potential for unintended consequences. Treating genetic diseases. Gene editing holds promises for treating and potentially curing a variety of genetic diseases by correcting faulty genes. Reducing childhood mortality. Human gene editing could be used to alleviate human suffering, improve quality of life, and reduce childhood mortality, improving health and agricultural productivity. Genetic technologies have the potential to improve health and agricultural productivity, longer and better quality lives. Some believes that these techniques could help people live longer and better quality lives. Potential risks and ethical concerns. Long-term effects. There are concerns about the potential long-term effects of germline editing, including off-target effect that might persist for generations. Designer babies and enhancement. The ability to modify genes could lead to the creation of designer babies or the pursuit of enhancements raised ethical questions about the fairness and societal impact. Ethical dilemmas. Ethical questions arise about who decides which traits are normal and which constitute a disability or disorder, and how to ensure equitable access to these technologies. Unintended, ununited consequences. That is unintended consequences. There is a risk of introducing unfavorable characteristics into an organism, such as antibiotic resistance or the production of toxins. Safety concerns. Genetic therapies are still new approaches to treatment and may have risks, including certain types of cancer, allergic reactions, and damage or to organs or tissues. Misuse and abuse. The legal, ethical, and moral boundaries of using genetic technologies are increasingly unclear, creating opportunities for their misuse and abuse. Limited diversity. Genetically modified species designed to be stronger and outlive other varieties could limit diversity within species. So far, we discussed about genetic modification for humans possible or not. We have done. Please subscribe our YouTube channel.